Welcome back to Archaeology After Dark, everyone. I am, of course, your host, Daniel Rhodes. My guest today is Danielle Irola. Danielle, thank you so much for joining me.
Thanks for inviting me to join.
Absolutely. So, this is take two on this episode. We had a couple of technical issues on the last time we tried to do this, but we think we sorted it out. First things first, I hope everyone had a great summer. We are back in business at Archaeology After Dark. So, Danielle, why don't you start off by telling us a little bit about yourself?
All right, so I am a PhD student. I specialize in biological anthropology and ancient DNA. I'm an archaeogeneticist. I'm currently working on some DNA analysis for two archaeological sites over in Poland, the vanished village of Getch and the former cemetery of Our Savior in what's now Cista Square in Wrocław. And then within ancient DNA, my specialty is infectious disease and identifying infectious pathogens in the DNA from human remains.
So, Danielle, what can we gain from studying a pathogen that's in a body that's no longer, I guess, has tissue on it? I mean, you can still get something from the DNA out of that, right?
Yes. Um, so what we can do is figure out, um, historically, we can kind of look at which diseases were causing epidemics in different places and different time periods and how those might have been spreading if we're finding, like with the plague, the Black Death, finding the same strain of the plague in different places. If we have different strains, looking at how closely related to each other they are, and that can give us some insight into where those strains were active and how they were changing over time to adapt to new environments and keep infecting different populations. Um, and so kind of looking at spread and stuff like that. We can also look at when skeletal remains have pathological changes in the actual skeleton, um, looking at like which diseases do we find in those individuals that might then be linked to those skeletal changes.
So you're saying you could compare a strain, like say the sites you're looking at in Poland, you could compare it to a strain, say in France or Italy, where it also was occurring, and you could see how the differences in those individuals affect the disease and how the disease infected those individuals also.
Yeah, we can basically build like a family tree or a phylogeny of different strains of the same disease. And that gives us some insight into how it was spreading and how it was changing.
Like how like different environments would change the disease. Like Poland, I assume, is colder than it would be in like say Italy or France.
Yeah, I think so. I mean, I did just wrap up about a month of field work in Poland, or 3 weeks, but it was there ahead of time, and it was really hot because summer.
Yeah, summer's hard.
Yeah.
So, what's your end goal while studying pathogens? I mean, I assume you can still gain something new from it.
Yeah. Um, so with the stuff I'm working with now, we're trying to figure out kind of in that same theme of skeletal changes and pathological changes in bones and which disease might have been causing that. Um, Broadly, there are some things about different diseases we're still trying to figure out. We're trying to figure out, other people are working on, the plague again and figuring out was that continuously spreading into Europe or was it hanging out in Europe and in some kind of unknown reservoir population and continuing to cause outbreaks. There's stuff like there are epidemics historically that we don't know what caused them. We have the historical records describing symptoms and stuff like that. We don't actually know which pathogen it was. Um, so there has been some work trying to do that with different diseases around the world and through time.
Yeah, and that's something I see on the news on either Google or something probably once a week is they found like an isolated case of like the Black Death or something in like a remote corner of the United States or something like in the very small corner of like Texas or something, and nobody knows how it got there. They're just like, oh yeah, it happens, and everyone else in the world is like, it shouldn't, but okay.
Yeah. Yeah, plague studies are coming out just constantly. I actually read a new one a couple weeks ago, while I was in Poland.
Yeah, and just about everyone I read, it says that it had like a new source, like, oh, it was gerbils or
Yeah,
not washing your dishware properly or something.
yep. Both of those good ways to spread diseases.
Oh yeah, yeah. Because I mean, think about it. In medieval Poland or France, Italy, they didn't have what we call hygiene standards. Like, they just left
Oh yeah.
dead people out in the street. It's totally fine.
Yeah, and sometimes that was just because the epidemic would hit the population so hard that you didn't have gravediggers and you didn't have a priest who could come in and do the appropriate, last rites or whatever rituals you needed to do when a person passed away, because either too many people had died or they had left town trying to get away from the disease. So there are historical accounts from something like the Plague of Athens where they're just like people, we just left them where they fell because we didn't have the capacity to deal with it.
Yeah, and in the context of history, for those of you who haven't seen like a plague standard or statistic recently, I think it's always been like three-fourths of Europe's population at the time, or like a projected population, because they didn't have like the census that we think of now. So it's just, a statistic of people who contracted it, I assume.
Yeah, that would be for the Black Death, which was the 1347 to about 1352, 53-ish plague outbreak in Europe. Like the statistics are wild, like just how many people are estimated to have died.
So when you're studying pathogens like this, you can, I guess, find evidence of treatment in it also, because I guess those treatments would be in the DNA at some point, wouldn't it? Like a vaccine. Well, I guess a vaccine would be out of the question because they didn't have vaccinations back then, but something like that's evidence of trying to treat it, maybe.
Not that I'm aware, because I think everything that and some of the historical disease treatments are, by our standards, quite wild. They aren't things that would get into the body cells and change the DNA. It wouldn't change anything at a molecular level. Um, so there's not really that. There are you can test for chemicals. I don't know how to do those because I strictly do DNA. But I think you can test for like mercury or something like that. I'm really not sure how that works.
So you are, I'm trying to how to say this. So you get permission to go to Poland to get the DNA samples out of the people. Yeah, you have to go through probably a lot of permissions to do that. It's um, an ethics thing here, there. How is it? I guess how's the community response to what you're doing?
To be honest, I don't really know. All of the kind of ethical and legal legwork has been handled by my colleagues over in Poland because they're the ones in charge of the sites and the human remains. And I haven't looked into what kind of legislation might be in place there. We do have like a formal agreement with them that lays out, okay, here's who is involved in the project, both the research institutions and the individuals. Here's what we're going to do and who's allowed to publish work about it. So in scholarly literature later, once we have more results and time to write those papers. But there's definitely it's going to vary so much on cultural context, because there are going to be groups of people who do not want DNA testing for potentially their ancestors or relatives, because DNA testing, as of right now, for the most part, it is a destructive process. So some people will not want you to go in and destroy human remains in order to get DNA.
And that's another thing. A lot of institutions are shifting towards a policy of not publishing research or at least advertising research that actually has imagery of human remains in it. Well, they'll still publish like the papers and stuff, but they won't show like the imagery of the actual remains because of the ethics that people are consistently shifting to.
Yeah. There's definitely like certain journals too where you can't have images of human remains, but then there are also other journals that where you can have images of human remains. I know the SAA's Society for American Archaeology takes it very seriously, and you have to let them know when you are registering for the conference, which I'm supposed to be doing right now. You have to let them know if your presentation, poster, or oral presentation, a talk, will include images of human remains.
And honestly, I think that's a great policy shift because they're taking the community's interest into the account of the research too. Like they don't want to offend the people who sponsor the work or who are actually benefiting from the
Yeah,
and then
yeah, exactly.
and then if you go on the internet, there are the people who are just way too into seeing the human remains. Like, so did you see any?
Yeah. Yeah, it's definitely I'll see on Instagram, just random photos of some individual skull, and I'm like, why do you have that? And there's often no context. So they won't explain to you why they're sharing this image of human remains. And for certain cultures, it's very taboo to view human remains. And that's why we have all these policies with our professional societies now.
Yeah, and they're like, so we have this skull. It's beautiful. Have you seen
Yeah.
it? It has all 32 teeth. All the teeth.
Yeah. That could be kind of rare archaeologically, having all the teeth still in there.
Yeah, that's true, but also, they want to display like the skull, like Vanna Whiting it, like, look at what we have.
Yeah.
Ooh.
And it's like, as much as I absolutely love looking at human skulls because I am trained in skeletal analysis, I feel like if we're going to share these images, we should be teaching something about it. Like here's what makes a skull interesting, what makes it important, what can we learn from that, rather than ooh, it's so macabre, so gruesome. That doesn't sit well with me.
Yeah, exactly. And that's why, again, I think so many people are taking a second look at policy changes about like displaying human remains. Like, oh, yeah, so this used to be so and so's granddad. We shouldn't display that.
Yeah. Yeah, because there's a question of not just do we display human remains, but whose remains are we displaying too?
Yeah. And, again, that's something that people are taking into consideration when, maybe a decade ago, people were just like, oh, yeah, just go ahead and put it in the display case.
Yeah,
This is fine. This is totally fine.
yeah. And again, like there is certain countries will have legislation that kind of guides this, and that is going to look different in different places.
So, shifting a little bit, you get your DNA samples from the people you've excavated, right?
Mhm.
You bring them back for analysis here in the United States, or is that analysis done in Poland?
I have done all my analysis here in the US. So kind of how this works is that uh, for skeletal remains, one of the best places to get DNA is in the teeth. So my colleagues over in Poland just sent me some teeth. They did get stuck in customs, I think, for a while. Um, but we get those samples out of the pulp chamber of the tooth because it's like a closed environment. It's protected from potential sources of external contamination, say the archaeologists who are actually excavating the remains, stuff like that. So we did this in the States because my university already has all of the lab equipment and everything set up. Um, we have all the protocols, we have the people, the faculty here to train us how to do this, and all the things that we need to help try and limit contamination of those samples. We did have to, once we had done the extractions and prepared everything for sequencing, we did have to send the samples I'm working on to a biotech company because they have the actual machines that can sequence it the way that we wanted it to. Because there are different ways to do that. So we needed the bigger, shinier machines that my university doesn't have at the moment.
Everybody likes the toys that like
Oh yeah.
it's not something we advertise in the field of archaeology to get people to sign up, but we do have probably the coolest toys. I mean, really.
We do. I would love to do a deep dive into all of the different machines and what they do, what their capabilities are, not just because it's good for me to have that knowledge for the rest of my career, but also it's just cool.
Yeah, like I want someone to take me on a lab tour and be like, "Ooh, what is this thing and what does it do?" Second,
That's part of our training is
second
we do a
question.
lab tour of our yeah.
Second question, can I touch it? Just touch
Yeah.
it a little bit. And then like, no, no, you can't. I'm like, oh no.
Yeah. But that's part of our training is we do like a lab tour of the genomics and genetics department on our campus.
Okay, that sounds phenomenal. And if it wasn't like 7 states away, I would definitely sign up
Yeah.
for that.
Yeah, and we're, slowly we can, depending on getting grants and funding, we can add to our collection of machines. We got one that does the extractions for us if we have enough samples to use with it. And I was like, damn, I just did all my extractions. It did it all by hand. The robot didn't do it for me.
I imagine that saves time as far as like analysis goes because you can just transition from like it's a streamlined process instead of having to do it all just by hand.
Yeah. I tend to do everything by hand just because when you're doing ancient DNA stuff, oftentimes your sample sizes are not large because first if you're working with an archaeological site, that's already a pretty limited sample size depending on where you are, and what does your burial site or your cemetery look like. um, and then you might need to narrow down that sample size further. um, so like from one of the cemetery sites that I am working with, actually, yeah, kind of both of them in a way. We're not sampling everybody. They're based on the questions that we're asking with the DNA analysis. That's going to kind of dictate, okay, well, exactly which individuals are we choosing for the DNA analysis? So for one site, it's the ones that have pathological changes in their skeleton. So since we're asking about pathogens, those are the individuals that we're going to sample.
So, can you tell what you're looking for by just like looking at a skeleton? Like, are there markers in like the bones just from like glancing at it, or are you looking like with a special device to find what you're looking for?
To an extent. So a lot of the pathological changes that we might see in a skeleton that are suggestive of an infectious or metabolic disease, metabolic being something like a nutritional deficiency, and then we wouldn't be testing DNA for that. Um, but one, these changes aren't always necessarily specific to a disease. So you can't always look at the skeleton and immediately go, boom, that is absolutely tuberculosis. Uh, Sometimes it depends on the, I guess, severity of the infection and which bones the disease is infecting. Um, and some of these diseases, and there aren't many infectious diseases that will infect and change skeletal material, but some of these diseases will also look quite similar to each other. And that is, again, where that prompt to do DNA testing comes from. And then when we do the DNA testing, there's a couple ways that we can do it for pathogens. Some you can test for specific pathogens, um, and that will then influence what lab methods and what machines you're using. Or you can do like what I did, which was we call it whole genome shotgun sequencing. And basically, we go in and it's like a spray of shotgun pellets, and it goes in and grabs whatever might be in the sample. It's not going to get everything, but it's going to get a lot, and it pulls that out. And then I can run that through different software programs to look at those DNA sequences and try to classify those sequences as belonging to a specific species.
So it's like casting a wider net.
Yes, exactly.
Okay. So
Yeah, because if you target specific diseases, you might miss something that you didn't have on your list and you didn't look for.
Okay. And that way you can narrow down your search results as you get further along. Like, say, from day one, your search would be smaller on like day 30.
Yes, because then you can run it through even more programs to look more specifically and kind of refine your analysis. And I've been working on that process recently.
So you are looking at the DNA, you're looking at the pathogens. Can you see how those pathogens have like eroded over time? Like, are the infections like less severe in like certain people, like say an older person versus like a younger person or something like that?
Not necessarily. I can look at the amount of sequences in each individual that's tagged as belonging to a specific species. Um, and that could just be down to how well the DNA is preserved, because the challenge with ancient DNA is that it is degraded. It's falling apart over time, and that's why it's fragmented and difficult to study. Um, but also, as far as what clinical symptoms the individual might have been showing at the time that they passed, that's something that I can't get out of the DNA. So because I don't have, most of the time we don't have soft tissue or anything like that. We'd have to look more at the skeletal remains, because that can kind of give you, for certain diseases, like an indicator of what that would have been doing clinically.
Yeah, I was just trying to think of what, diseases, don't start off with like a physical symptom like a cough or a skin lesion or something. That's not something you can get from someone's skeleton. If a skeleton is coughing, that's
Yeah,
time to leave.
that's cause for concern.
That's cause for packing up and leaving like immediately.
Yeah. Yeah, as much as I do enjoy working with human remains and I find it so fascinating, that's not really something I feel the need to experience.
Yeah, I feel like like I've seen one too many movies to not like ultimately jump to like the worst case scenario while immediately looking at human remains. All right, so I need to plan in case this thing starts to move while I'm studying it. That would be bad.
Yeah.
Did that toe just twitch? Uh-oh. Time to go.
Yeah.
Let's go.
Yep.
So I assume you can see how the pathogen has changed. We've talked about this. It like varies between regions. Also, like we talked about this during the last recording. We compared this to COVID, how there was like a new strain every so often, and you can see this in individuals that you're looking at also because a disease changes over time because we also change it as people.
Yes, we can look at when and where are our species acquiring different mutations. How are their gene variants changing at a molecular level, and we can look at those and look at where they are in the world. So something like tuberculosis is split up into multiple different lineages, and there are lineages that are more commonly found in different regions around the world. So, for just a specific example, what's called lineage 4 is most commonly found in Europe, um, stuff like that.
And that would be different from the tuberculosis strain that were prevalent here in the United States for the longest time. I probably should know that time frame.
Yeah, the evolutionary history of tuberculosis is kind of very, very complicated. But I think lineage 4 might actually be very common in the US. I think it might have. Mm, that one I'm not so sure about, but it, they'd still be very, very closely related because they are the same species. Um, but it would just be more closely related to like other lineage 4 strains, if that makes sense.
Yeah. And I guess that's why we have to consistently study these diseases also because some of them, are still here. And again, we get the random pop-up cases of these diseases. Can what you're doing help us like create new vaccines or treatments for them?
I
Yeah.
think that's one of the ultimate goals, is that, or how to prevent them at all. And I think it kind of leads into the concept of personalized medicine, but kind of the other way around, because with personalized medicine, we're looking at human DNA when you're really sick and you don't know how exactly to treat it. Um, we can look at what are your genes doing? What are the what are the proteins that they're coding for? And what are those going to do in your body? And then based on that, how is that going to interact with different drugs? So I think with disease, the goal is to kind of do that, maybe not necessarily in reverse, but like look at what is this disease or this strain of that disease doing at a molecular level, and how might that react and interact with different treatments. And this would be super important for something like tuberculosis because it is increasingly very, very drug resistant.
Yeah, and that happens a lot with diseases. Like we talked about with COVID, like again, there was a new strain every so often, and we still have to come up with a new vaccine just to change. And it's always like a regional variant that has
Yeah,
adapted to like the climate of the region it's in. Like, say, if it was in somewhere that's colder, like Russia or Siberia, it would be more resistant to the cold or something like that.
yeah,
Because I totally know how to treat diseases. I'm 100%
yeah,
skilled.
yeah. I do read a fair bit of like clinical literature about how do we treat this and what is it looking like in modern patients. Um, but I'll definitely come across stuff on modern websites talking about how because they're going to talk about the disease in modern populations, and I'm like, what you're saying here does not apply to 1600s Poland.
No, I'd like to think people have changed since the 1600s, but
Yeah,
I can think of at least a dozen people who tell me I'm wrong. Say
yeah,
that. No, the people haven't changed. We've always been people, and to say that we have changed is ludicrous.
yeah.
So, what is your ultimate goal? Like, why study this as a field of study?
I honestly just find it fascinating, and I ended up studying human remains because I wanted to be an archaeologist as a kid. Um, but I also couldn't decide where in the world do I want to do my archaeology. Did I want to do Roman archaeology? Did I want to be an Egyptologist? And then I started studying human remains, and I was like, one, this is fun. I really find this so intellectually engaging because every skeleton can tell a story. Um, but then I was like, oh, I can take this skill set, skeletal analysis, anywhere in the world. I don't have to limit myself. Um, and then so went started going the bioarchaeology route, and then got into my PhD program where we heavily emphasize and do ancient DNA. And then I was like, oh no, this is the most fun. So it's kind of has given me a place within archaeology without having to actually be an archaeologist.
And that is one thing that I don't think people like grasp about archaeology until they really get into it. It's something that you actually can do anywhere. Like
Yeah,
you need to get permissions for everything because again, we're circling back to the ethics thing here is you can't just randomly go digging up people. That's highly
yeah,
illegal and highly unethical. So please don't do that.
yeah,
That's my one PSA for this episode.
yeah.
Please don't
Mine would be
please don't exhume people without permission.
oh oh yeah, you need permits for that. Mine would be if you find human remains in the woods, call the cops, please.
Yes, and it's fine when you say like an archaeologist said they found human remains, but if your neighbor tells you they found human remains, it becomes a different situation altogether, and you're just like, oh,
Yeah,
I need to leave, I need to move, I need to relocate.
yeah,
So
yeah.
if people wanted to learn more about ancient DNA or the process of studying it the way you're talking about, what would you recommend to them?
I'd recommend a couple things. Little shameless self-promotion here. I create content about ancient DNA and science in general and archaeology on my Instagram and my YouTube channel, which is called Considering Skeletons. Um, but I would also recommend the book Neanderthal Man by Svante Pabo. it is very much his memoir, but it is he's kind of like the guy who was he's like one of the founders of the field of ancient DNA, working on Neanderthal DNA from the 90s onwards and establishing the labs and the protocols and figuring out who do you need on your team to make this happen? What kind of skill sets do you need to bring into this to get the data and analyze it?
And I mean, honestly, just thinking about that is fascinating. Being the person to like establish the protocols for something like, oh, no one actually has a process for handling
Yeah.
human remains in this capacity, so I guess I should probably be the one to do that.
Yeah, he's definitely not the only one who is involved. He did actually win a Nobel Prize in medicine for it, which is kind of crazy. I'm like, anthropologists, we don't get Nobel Prizes, but he did. But also, like, I will sit in our lab and just wonder, how in the hell did anyone figure out how to do this?
I mean, I have that like epiphany on occasion too. Like when I'm working with somebody who's been doing this like way longer than I have, like I'll just hold up this little piece of a thing and they're like, oh yeah, that's so and so from like the style of the late 1700s, early 1800s. And I'm just like, what?
Yeah, I have that same experience because like when I study skeletons, I'm usually looking at adult skeletons. So then if I get the chance to work with and learn from someone who knows subadult and juvenile skeletons, which obviously look really different because they're still growing and developing. And they'll look at like some little fragment and be like, "Oh yeah, that's this. It just looks like this because it's it hasn't fully formed yet." And I'm like, "Damn, I didn't see that at all."
And then there are the people who study, like animals, and they're like, oh yeah, that's from a raccoon.
Oh yeah, I know nothing about zooarchaeology, animal remains. I've worked with a comparative collection, like mostly inventory maintenance, um, stuff like that, some cleaning, but like I'm not gonna know anything looking at an animal skeleton. Like not my skill set.
Well, Danielle, it has been a lot of fun having you on this episode. Thank you for joining me. And we've got the technical issues worked out from the first time, which is great.
Yes. And again, thank you for inviting me. This has been so much fun.
Yeah, absolutely. So y'all make sure to check out Danielle's stuff. I'm going to put links for it in the description, and her Instagram also will be in the description. So feel free to check that out at your leisure. And again, welcome back from summer. Bye, everybody.
Bye bye.